to have some very important guests to come and give a address. Fortunately, our principal is not. To tell you something, why McGill is so well known in research. You see, she's our principal, and yet before this, she was the president of the Natural Science Engineering Research Council that look after all the granting of Canada in this area. And before that, in Queens, she was the vice principal of research and also vice principal of administration. And furthermore, she was professor in chemistry and computer. And of course, she graduated from McGill. So without further ado, I would like to invite Dr. David Andelman, is Vice Principal, Health Affairs and Dean of the Faculty of Medicine, McGill University since 2012. He received his MD from McGill in 1979, then completed internal medicine training at the University of Toronto, and then trained in respiratory medicine at McGill. He joined McGill's Department of Medicine and began his research career at the Milken Christie Laboratory on the pathophysiology of chronic inflammatory disease of the lung, particularly asthma and COPD. Before becoming dean, he served as director of the Division of Respiratory Disease, chair of the Department of Medicine, and physician-in-chief of the McGill University Health Center.
Merci, Dr. Chang. Bonjour et bienvenue. J'ai l'honneur de vous accueillir ici au nom de McGill et de sa faculté de médecine. Good morning, everybody. I'm delighted to be here today to help celebrate the 60th anniversary of the invention of artificial cells and the contributions of scientists at McGill and from around the world. I'm very pleased to be able to be here together with the esteemed consul and head of Office of Education, Science and Technology and Culture from the Chinese Consulate, Sheng Xiang, Quebec's own chief scientist, Rémi Quirion, distinguished keynote speaker, Professor Zhang and his team members from Peking University, our guests and alumni from here and abroad. I extend very warm greetings on behalf of McGill's Principal and Vice Chancellor, Suzanne Fortier, who, as you just heard, could not join us this morning, even though she was looking forward to being here. But I'm sure she's very pleased to see this excellent turnout. We're very fortunate to have a history of excellence in physiology at McGill. And thanks to strong faculty members and leaders such as Professor Thomas Chang, the amazing person who you just had speaking to you, and the department's chair, John White, who you'll hear from in a few minutes, who've been able to prepare future generations of researchers to help battle some of the most pressing challenges facing humans and their communities. So I referred to Professor Chang as amazing. I think he's sort of a living legend. He's really a trailblazer ahead of his time. In 1956, while still a physiology undergrad here at McGill, his scientific curiosity helped him to invent artificial cells in a makeshift lab in his dorm room. Equipped only with a perfume atomizer, some colloidin, and hemoglobin borrowed from a lab, Professor Chang managed to build a 1 millimeter round plastic cells that contain hemoglobin. He published his first scientific paper in Science in 1964, and in 1989, New Scientist called his student research project an elegantly simple and intellectually ambitious idea that has grown into a dynamic field of biomedical research and development. His original ideas were years ahead of the modern era of nanotechnology, regenerative medicine, stem cells, gene therapy, and blood substitutes. And now 60 years later, Professor Chang has earned his academic stripes many times over. He's currently Emeritus Professor of Physiology, and he's the longtime director of McGill's Artificial Cells and Organs Research Center. He's a member of the Order of Canada, and evidence of his stature in the international scientific community is shown by the fact that he was nominated for the Nobel Prize twice. On a more local level, during our 190th anniversary celebration a few years ago, Professor Chang earned a title amongst the greatest McGillians. And he's been called the Jules Verne of artificial blood research and the red blood cell man. His drive to continue advancing science and improve the health of populations continues strong, as evidenced by this symposium. To quote Professor Chang, To me as a scientist, what is most important is what is most useful to the patient, not is what is good for your reputation or what pays you the most money. The sick patient should be the most important stimulus for your work. So, Dr. Chang, on behalf of McGill and the populations and communities that we serve, thank you very much for the commitment and dedication to improving healthcare around the world that you've shown. And for inspiring your students, including your first PhD graduate who will address us shortly, Emeritus Professor and Director Mark Poznansky from the Robarts Institute, who will really show how Professor Chang has allowed greatness to go from generation to generation. We look forward very much to hearing your anniversary lecture, Professor Chang. Alors, je vous souhaite à tous un symposium intéressant et enrichissant. Merci beaucoup.
Thank you, Dean Alderman. Thank you for all the kind words. So, may I introduce now Professor John White. He's the new chairman of the Department of Physiology. He received his PhD from Harvard in 1986. His postdoctoral work focused on the fundamental mechanism of mammalian gene transcription. He has been a member of the Physiology Department at McGill since 1991. Since 2017, he is professor and chair. Since coming to McGill, he has made important contributions to our understanding of the role of vitamin D in human physiology. His research opened the field of study of vitamin D as an inducer of human antimicrobial immunity and has provided fundamental insight into the mechanism of its action as an agent of cancer prevention. Professor White.
Well, thank you, Tom, for the opportunity to speak here today. As Tom mentioned, I am a recent chair of the department. It's been a big year. I turned 60 this year, just like artificial cells. And I have to say that artificial cells are looking much fresher than I am. As Dean Eidelman mentioned, the Artificial Cells Project started as an undergraduate project in the physiology department. If I understand correctly, Tom was one of the guinea pigs in that respect. He was one of the first 4 students who did an undergraduate honors project in the department. And that was an initiative of Hank McIntosh, who was chair at the time and probably one of the more dynamic chairs in the history of the department. And after he got his MD, Tom turned artificial cells into a PhD project. The problem was, who's going to supervise it? The project was so avant-garde that really the only expert in the world was Tom himself. I gather that Hank McIntosh agreed to supervise the project, perhaps with some reluctance because it wasn't exactly his area of expertise, but obviously the project worked out extremely well. And as you are all aware, it turned into a single-author paper in Science published in 1964 that has now been cited 1,200 times. The rest, as they say, is history. Tom became a faculty member in the Department of Physiology in 1966 and rapidly became a full professor. And in 1979, the Center for Artificial Cells and Organs in the department was started. And since then, of course, as everyone in this room knows, it has become an international network. The journal that grew out of it, Artificial Cells, Nanomedicine, and Biotechnology, has been ranked by Reuters as 4th out of 77 journals in the field of biomedical engineering. I am very proud to be chair of the physiology department, and I'm even more proud that Tom Chang has been a pillar of that department since 1966. I really think that this symposium is a fitting honor to his scientific legacy, and I wish all of you a stimulating and thought-provoking time. Thank you very much.
So now I want to introduce Mark Poznansky. Mark Poznansky recently retired as director of the Robert Research Institute of the University of Western Ontario, and he has been a full professor there also. He's a Montrealer and was a physiology undergraduate at McGill. He was so bright that one of the professors was very upset with him because he kept asking him questions that he can't answer. So And he worked as a project undergraduate student with me, then became my first PhD graduate student. He worked hard and he could work with both hands simultaneously for some reason. So he worked hard and he finished all his research project in half the time and completed his required PhD research in 2.5 years. So he has to sit around for another 6 months to qualify for the 3 years requirement. He then spent his postdoc at the laboratory of the head of biophysics at Harvard. He wanted to come back to McGill, but at that time, there was no position in this area for him. So he became associate professor at the University of Alberta at Edmonton and quickly became full professor, then vice dean of research. He was so good that he was recruited by the well-known Robert Research Institute of the University of Western Ontario as their director and full-time full professor. He held this position for many years until his retirement recently to become president of his own consulting firm. He held many important national and international board positions, including as director of the Ontario Genome Board. He is also a member of the Order of Canada. Mark?
Professor Chang, guests, it's an honor to have been asked to represent the hundreds of graduate students, postdocs, and trainees of the Chang Laboratory over the past 53 years. In 1963, as a young undergraduate student in physiology at McGill, I had a dilemma. With little knowledge, I had to choose an undergraduate supervisor, and the pickings were significant, as it was a large, active department with 3 fellows of the Royal Society of Britain, F.C. McIntosh, B.D. Burns, and A.S. Burgeon, and a host of well-trained and well-funded young investigators. And then there was Tom Chang, a newly minted MD PhD, working on artificial cells. He wanted to create completely artificial entities to replace biological cells that were either absent or not functioning. He wanted to create artificial blood to be used in place of transfusions, especially in areas where fresh blood was difficult to obtain. He wanted to produce cells that could help detoxify blood in patients in kidney failure. Now, realize for a moment that in 1963, the structure of DNA had just been announced, and there was no such thing as genetic engineering. The first stem cell papers had been published, but really only as a concept. High-risk organ transplantation had started a few years earlier, and regenerative medicine was not yet on anybody's lips. But this young McGill faculty member's enthusiasm was contagious. He had a vision, and he had a pretty good start. He succeeded in creating semi-permeable aqueous microcapsules, which in essence could function as artificial cells to mimic red blood cells if they were filled with hemoglobin or filled with a particular enzyme for the potential treatment of enzyme deficiency diseases or inborn errors of metabolism, the project that Professor Chang assigned to me as an undergraduate. Now, if the truth be told, at least in Canada, Tom's ideas were met with a huge amount of skepticism. But he had, and this is very important, he had a few senior people who believed in him and gave him a break. There was Hank McIntosh, a huge force in Canadian and British science in the 60s, who was chairman of the department at the time and had been Tom's thesis advisor. And there was Sir Malcolm Brown, president of the Medical Research Council of Canada. Today, we understand the extent to which Tom was at the cutting edge of developments in biotechnology and nanotechnology, except for the fact that those terms or disciplines had not yet been defined. Skip forward a half century, Professor Chang is still in his lab at McGill working on the use of artificial cells to solve particular problems in medicine. But now the world's caught up to his revolutionary ideas, and the entire area of nanotechnology represents one of the most exciting technologies of the early 21st century, not only in medicine, but in any number of other industries. By 2010, the entire genome had been well defined, and the genomes of many different organisms had been sequenced, and many technologies associated with genetic engineering, including gene editing, had been demonstrated. And the entire area of artificial cells, championed by Professor Chang in the 60s, was no longer a dream or some sort of futuristic objective. In 2011, Craig Venter, one of the major players in the Human Genome Project, announced that he had created or manufactured a completely synthetic line of live cells. The DNA code came from his computer. A long strain of DNA containing the necessary genes was synthesized and placed into the empty shell of a bacterium, from whence it proceeded to both replicate itself and create the proteins necessary to sustain life. With these cells, Venter anticipates being able to produce cell lines that create vaccines or specific medicines or anti-cancer therapies or even nitrogen-fixating bacteria that can be used for the replacement of expensive and sometimes inefficient fertilizers. With the use of modern genomic and computer technologies, Craig Venter had succeeded in creating the same artificial cells with the same objectives that a young Tom Chang at McGill had proposed and developed, albeit with much less sophisticated technologies, some 50 years earlier. In every sense, Tom is a father of not only artificial blood substitutes, but really the entire area of nanotechnology. Finally, from a personal comment about Tom Chang, as a teacher and as a mentor, even as a newly minted professor, he had a sophisticated Socratic teaching method. I'm not sure he was a student of Socrates, but at least he used the technology. Late in the afternoon, we would be discussing a research problem, and he would challenge me. He would say, I can think of 4 or 5 ways of going about solving the problem. And without divulging any of them, he would say, let's discuss them in the morning. I would spend the evening thinking about them, about his 4 or 5 solutions, and in the morning we sat down to discuss, and maybe I would have figured out 2 or maybe 3, and if I was lucky, and on occasion I even thought up of an original one. This was what it was like working under Tom's tutelage, and on behalf of all his trainees, I thank him. Thanks, Tom.
Thank you, Mark, for all the talk about your experience. Okay, the next presenter, it's a pleasure to introduce Professor Remy Quillon, OC, PhD, FRSC, CQ. He is the first chief scientist of Quebec, which means that he looks after all granting agencies in the province of Quebec, which is a very big job. At McGill, he is a full professor in psychiatry. He was vice dean, Faculty of Medicine, and a senior university advisor in health science research. He was also the scientific director of the Douglas Mental Health University Institute at McGill, and under his leadership, it became a premier research facility in Canada in the field of neuroscience and mental health. He has published 5 books, more than 650 papers and articles. He was also the inaugural scientific director of the Institute of Neurosciences, Mental Health, and Addiction. He received many awards, including the Special Royal College Fellow of the Royal College Society of Canada, Royal Society Academy of Science, Chevalier of the Ordre National de Quebec, and a member of the Order of Canada. Professor Quillon.
Merci beaucoup, Tom. C'est un plaisir au nom du gouvernement du Québec d'être avec vous ce matin. So as was mentioned by the colleague, Dr. Chang is a real pioneer, like you will not meet so many times in an academic career. For me, I was at McGill for close to 30 years. Tom Chang was at the comparable to a household name like Brenda Milner For those of you that know neuroscience a little bit, Brenda Milner, pioneer in learning and memory. She will turn 100 in a few months. She's still active. Charles Taylor, a philosopher at McGill University, and Tom Chan, a superb trio with international reputation and leadership for almost 60, 65 years. Today, as a chief scientist of Quebec, as was mentioned, I oversee the activity of our three funding council in health, in science and engineering, in social science and humanities. I think that Mark already mentioned how far ahead Tom was. One of the things that we're trying to do at the Quebec funding agencies is to make sure that for a huge challenge of our society, like building artificial cell, we have experts from many disciplines working together. So almost 60 years ago, Tom was already doing that. But today, it's still a challenge for many scientists in academia to work together on very significant problems. Just think of climate change. Just think of aging. Yes, you need healthcare expert, but you also need engineer, you need chemists, and you need social scientists. So one of the recent programs that we developed at the funding agencies is called Audace. So to have audacious, very far-thinking program, having experts from different disciplines applying for grant. And if I use a baseball analogy, not to get to first base, but to have a home run. So I think that Dr. Chan should apply for one more grant from us in this type of program, and I'm sure that he'll be very successful. Merci beaucoup et bonne journée.
Thank you, Professor Quillon. It's a pleasure to introduce Xiang Chang. The reason I put him at the end is because later after I talk, we have a special award from the journal for somebody from Beijing's group. So I welcome him to come and say something. And he is the counsel and head office of Education, Science and Technology and Culture of the Consulate General of China in Montreal. He graduated from Jiangxi University, China in 1992. For 5 years, he was program officer of the university in internal collaboration and exchange. In 1997, he spent 1 year as a visiting scholar in Budapest, Hungary. 1998, he, for 3 years, was program director, then deputy director for another 3 years for the Chinese Service Center for Scholarly Exchange, Ministry of Education, China. 2004, for 4 years, he was a counsel of Education Office, the Consulate General of China in Chicago, USA. 2005, for 5 years, he was the director of the Chinese Service Center for Scholarly Exchange, Ministry of Education, and since 2014, he holds his present position. Dr. Xiu, please.
Distinguished guests, ladies and gentlemen, good morning. On behalf of the Consulate General of People's Republic of China in Montreal, I would like to take this opportunity to extend my heartfelt congratulations to Professor Chong. I would like to also take this opportunity to extend my cordial welcome to all the delegates, including overseas Chinese and Chinese delegates. 2 years ago, when I came to Montreal to work at the Consul General, I worked as Counsel for Education. my predecessor, Mr. Zhai, told me there is a very famous professor in McGill. You should meet him sometime. When I first met Professor Chong, I'm deeply impressed by him. I'm deeply impressed by his spirit in research. So when I go to McGill, every time I tell the Chinese students in McGill, today you are proud for McGill. Tomorrow, McGill will be proud for you. You should learn from Professor Chong. So I'm very proud Professor Chong made so outstanding achievements. I would like to take this opportunity to extend my heartfelt congratulations to Professor Chong. Congratulations. 今天呢，在座的有很多来自中国的专家学者，那我很高兴的借此机会代表中国驻蒙特利尔总领馆，向大家呢表示热烈的欢迎，向张教授表示衷心的祝贺。我们为有这么一位优秀的华人专家学者感到呢非常的自豪。我希望所有的中国学者能够像张教授一样取得更好的成就。So, because artificial cells is so new to me, I have nothing to say except my cordial thanks, my congratulations, Professor Chong. I hope every delegate make great achievements like Professor Chong. Thank you so much.